The way this class works is a little bit funny, and if you look at the schedule, you might be a little bit confused. We meet Monday, Tuesday, Wednesday, Thursday from 9 to 9:50, and then Wednesday from 9 to 10:50, okay? Wednesday is a lab period, so if you have an 11 o'clock class, you're going to have to kind of be careful to make sure you get wrapped up so you can get to your 11 o'clock class. Friday is also a lab period, okay, because a lot of times we will inoculate cultures on Wednesday, and then you need to come back and look at results on Friday. So Monday, Tuesday, Thursday will be the traditional lecture sections, and every class will meet in here, both lecture and lab. And then Wednesday we'll have lab, and then Friday we'll have lab again. This first week we're not having lab. We're just not quite ready to get involved in it. So what I'm going to do is Monday and Tuesday, today and tomorrow, will be the traditional lecture sections. And then Wednesday, I'm going to use half of the 2-hour period for another lecture. And then Wednesday, the second hour, I'm going to kind of orient you to lab. Okay, and so make sure you understand what we're going to do, what the philosophy of lab is. You can sit wherever you want to for lecture, although people tend to find a seat and sit there throughout the course of the semester. But when you come in Wednesday in lab, I'm going to ask you to stay there the rest of the semester because we're going to assign you lab drawers. You will be doing some work in pairs, and so if you have someone you really want to work with, you might want to try to sit by them. If you have somebody you really want to avoid, you might want to go on opposite sides of the room. There's a lot of free interchange in lab and a lot of moving around and things, and so it's not that big a deal if you get stuck next to someone you're not normally used to working with. Working in pairs in labs oftentimes just involves sharing cultures and inoculating some media, and your partner will do part of it and you'll do the other part of it. But lab is pretty freewheeling. This is the only class that's taught here this semester, and so the lab will be open every day from 8 to 5. I typically don't keep it open on Wednesday, on weekends or evenings, unless some things are starting to run up that are really causing you to need some extra time, and we can probably find some work study students to proctor if we need to do that. You will be coming into the lab a fair amount on your own. That's just the nature of the beast. Some labs you will finish before the 2-hour period is up. And if you get your work done early, I certainly don't mind if you leave as soon as you're done, because you will pay me back more than enough in terms of hours you spend in lab. When you're working on your unknown, for example, there are times when you're going to have to come in and do some work out of traditional class period. And like I say, you can come in anytime during the day. That's not a problem. Okay, let's see what other... We'll say more about lab on the Wednesday session that we have. Any questions? I apologize for the crowdedness of this. We try to normally keep this class at 15, so we don't have to have anybody sitting on this row, because as you can see, it's pretty difficult to make it up and down these aisles. That's going to create a little bit of a problem when we do labs related to microscopy. And if you're sitting in this interior row and have questions, I'm going to have to kind of battle through there. It's a little bit more of a challenge for me this semester. Knee replacement 5 weeks ago. And so I'm getting around pretty good, but still hobbling a little bit. So we'll see how that goes. For class, most of the stuff's going to be directed either here or here. And so you're going to have to kind of figure out how you're going to situate yourself and where's the best place to sit. If you can bring laptops or other devices for the PowerPoint slides, that gives you a little bit more flexibility in terms of not having to look over here. But I will be using a combination of the board and the PowerPoint slides. PowerPoint slides are primarily images. I don't have a lot of text PowerPoint slides. Most of the text I will put up here in my bad handwriting on the blackboard. If you have trouble reading what I write on the blackboard, please ask me. Please let me know. Because as the semester goes along and as I get older, my handwriting gets worse and worse and worse. I'll probably go, man, I don't remember being that bad in cell biology 3 years ago. Okay? All right. So what are we going to do today? Well, one of the things that educational research clearly indicates is that Students are much more interested and engaged in a course if they believe that it's relevant. This is an elective. Now, many of you are taking it because it was either strongly recommended. If you're a pre-med, it's highly recommended. Some of you have to have this course. If you are a potential PA student, for example, you've got to have micro. If you are a pre-vet, you've got to have micro. If you are a pre-pharmacy student, you've got to have micro. So I've got a captive audience to some respect, but there was some level of choice for some of you in whether you took this course. I'm not sure why that was for some of you. Hopefully it's because you heard it was something that was interesting and possibly useful. But I'd like to spend a little bit of time today to start talking about the relevance of microbiology. Why should we spend a whole semester devoted to this particular topic? As we do that, we'll talk a little bit about what kind of organisms we're going to be studying. When did microbiology develop? I like history of science, and one of the things we're going to do at the beginning of this class is talk a little bit about the history of the evolution of microbiology. It's not a science that just all of a sudden, bing, it was there. It had a very interesting history, and it evolved over time. And some information about where are microorganisms found. That can be relevant when you do lab work, and particularly when you're working with your semester-long unknown assignment, because you don't want to contaminate your unknown and identify a contaminant. And so are there a lot of microorganisms around that can potentially serve as sources of contamination? As it turns out, yeah, microorganisms can live just about anywhere that you might conceive. So, relevance. Why are microorganisms relevant? I'm going to ask a lot of questions today. And one of the things I'm going to do, I'll just give you an aside, is I really like to, whenever possible, and there are a lot of occasions to this, is to relate this back to some of the things you have in cell biology. So you're going to find me saying many times, we covered that in cell. Do you remember that? Some of you who had last semester, that'll be a little bit easier. Some of you will have to dig back and think, wow, 3 years ago. Where was my head 3 years ago? So why? What's the relevance of microorganisms? Why bother studying them? Pardon me? They cause disease, and that's one of the things that people most associate with microorganisms are bad, detrimental activities. They make you sick. Okay, that's something we will talk about. Are all microorganisms bad? No. What if you had the ability to all of a sudden be Dr. Evil, and you had the ability to eradicate every microorganism on this planet? What would be the consequences of your action?
Probably not a lot of things would work anymore.
Absolutely. Life as we know it could not exist. Microorganisms are absolutely essential for the functioning of life on this planet. One of the articles that I'm going to post for you is an article that talks about our emerging understanding of the association that the human body has with microorganisms and all of the impacts that has on human beings. If you look at total number of cells in your body, you are 90% microorganism. Only about 1/10 of the cells of this body are actually human eukaryotic cells. The other 90% prokaryotic. Now, in terms of mass, they're so small that the mass of those cells is fairly small. That's an interesting insight, isn't it? I am mostly bacteria. And things like your nutrition and your mood and physiological processes, we are finding increasingly are being governed by microorganisms that are part of this body and not your eukaryotic cells. There are links to depression, for example, and your microbial population that makes up the majority of your body. Or obesity, or susceptibility to certain diseases. So I'm going to post that article quite soon, and there are probably several follow-up articles, I run across these all the time, that talk about this relationship between microorganisms and the functioning of the human body. Well, we're going to delve into some of these other things about relevance in terms of what bacteria do, good and bad, in a minute. But there's another relevant area that should be of interest to you. Jobs. There are all kinds of microbial-related careers in science, and the numbers of those is exploding and getting bigger and bigger all the time. And that's what this first slide that's up here tries to illustrate, is just some of the kind of microbial-related careers that are available. And a really big one that's emerged, that we'll talk about a little bit more later on, is biotechnology. All of this biotech stuff. Anybody had Morgan McComb's biotechnology class? Nope. She's going to offer it in spring, and you should take it. All of the applications of microbial metabolism and genetic engineering to human society have just transformed our society. I mean, look at Eli Lilly. Eli Lilly, how much money they made on being able to make human insulin via genetic engineering, and all the drugs that are being made these days by genetic engineering. So, life couldn't exist without these microorganisms. That's pretty relevant. And real career opportunities. That's pretty relevant too. Okay. So let's take a look at the what for a second. What kind of entities are we going to potentially encounter in a microbiology course? What's the key word there? Microbiology. Microbes. What are things that are microbes? What characterizes them? What does micro mean? Something is microscopic. It's small, right? So we're going to be dealing with tiny things, most of which you can't see with the unaided eye. There are a few macroscopic organisms that have fallen under the realm of microbiology. But traditionally, we're talking about microscopic things. So give me some examples. What are some examples of microorganisms? Bacteria? Okay. What else? Think back to your biology career, and what if you had to pull out the microscope to study? So that's bacteria. Anybody have botany yet? Have to use a microscope in botany? Yeah. I mean, you have to use it to look at plant tissues and things like that. But weren't there some algae that you looked at? What about an O'Neill zoology class? Use a microscope to look at anything? Look at any protozoans, for example, or any of the little ciliates or things like that? Go back to botany for a second. I don't know if you looked at any microscopic forms of this, but you certainly looked at some macroscopic forms of fungi. And there are some microscopic fungi. Probably one of the most infamous one is a little microscopic fungi that caused the Irish potato famine, which resulted in a net emigration of Irish folks to the United States and completely changed the culture of this country, all due to a little single-celled fungi. What else is really small? That might be the province of microbiology. Talked about it a little bit in cell biology. Agents of disease. Virus, okay? Well, now we're getting into a little bit of a nebulous area here, because do we consider virus to be alive? Typically not, okay? In cell, I try to make the distinction that viruses have characteristics of living organisms, but they only show those characteristics when they're inside of another living cell. And there are things viruses can't do, okay? They don't have both DNA and RNA, for example. They don't have ribosomes, so they can't make protein. So viruses fall in that kind of gray netherworld. Nevertheless, if viruses are in a curriculum, sometimes they were included in a microbiology course. So, which of these are we going to spend most time on? Well, as I said, we tend to cover the algae and botany. We tend to cover the fungi and botany. We tend to do those protozoans, and we do some in 134, but we tend to do them in zoology. Virus, I wish we had a virology class, but what little we talk about in virus or in cell. So what's left for my course, for this course? Pretty much bacteria. And in some respects, it probably would be more truthful to call this a bacteriology course than a microbiology course. There's one other thing we're going to spend a significant amount of time talking about in this course, and that is immunology. And we'll do it in the context of microorganisms cause disease. At least some of them do. And when you're exposed to these disease-causing microorganisms, your body responds with a protective mechanism, your immune system. And so we're going to talk about immunology in that context, probably about somewhere between a quarter and a third of the course. It has to do with this application of microbiology, immunology. And to be honest with you, we do that because we need to put this somewhere. There are certain disciplines, for example, if you're in medical technology, you've got to have a significant immunology component. So we say we do that in microbiology. And if you're going into any of the medically related fields, you need to know something about immunology. So really, from a practical point of view, this course really is a bacteriology/immunology class. So we're really not going to do anything with virus, protozoans, algae, fungi, etc. And when we do work in lab, it will all be with bacteria. Who developed the first formalized system of classification of organisms? I know Huygens told you about this in botany. Swedish guy by the name of Linnaeus. You remember Linnaeus? Carolus Linnaeus. And he developed a classification system that put all living organisms that he knew about at the time into two major groups. What were they? This was in the 1700s. What do you suppose a good bet was for the two major groups that everything fell into? Didn't know about prokaryote and eukaryote yet. Okay. No electron microscope yet. If you were a living organism, you were today either a... What were we? Animal or plant? Yep, plant animal. Or in Latin, flora and fauna. Flora plant, fauna animals. Well, this particular diverse group of organisms, plus all the plants and animals, it was kind of hard to put them into one of two categories. And particularly when they discovered bacteria. Remember bacteria? Is it a plant or is it an animal? Does anybody know where it was put under the Linnaean classification system? It was put in the plant kingdom. And we still today, and I will use this all the time, will refer to things like your bacterial flora. That refers to bacteria that inhabit your body, your gut, for example. And so that is the origin of that particular descriptor for those types of bacteria, as it goes all the way back to the Linnaean system of classification. Well, in the 1900s, the Linnaean system of classification was deemed to be just too simple to really accommodate all the diversity of microorganisms. Because there were some things that just weren't clearly a plant or an animal. So there was a guy by the name of Whittaker who developed a new system of classification that broke all living organisms down into 5 kingdoms. And this is what I grew up learning. This is what I grew up teaching most of my career. And it really has only been within the last decade that this system of classification has been replaced by another system of classification we'll talk about in a minute. You all familiar with the 5 kingdom of classification? Do you know what the hierarchy is in terms of breaking things down according to groupings? Kingdom is the highest level of classification, most inclusive. What's the next level below that? Remember? Kingdom, what's next? Phylum. Botanists are kind of rebels. What do they call phylum? In botany? They don't call it phylum. In animal, they call it phyla. In botany, they call it divisions. Kingdom, phylum, division. What's the next one? Kingdom, phylum, class, order. One before genus, family, genus, species. So what were the 5 kingdoms?
Animal, plant, fungi,
Animal,
bacteria,
plant, fungi, bacteria were in a division that's had several names. It's been called Monera, it's called prokaryote. That's the 5th one. Protista. A little protozoans and things like that. 4 of these kingdoms have something in common. What do they have in common? It has to do with their cellular structure. Sarah referred to it earlier. Four of them are eukaryote. They're all made up of eukaryotic cells. Which one is it? This kingdom is prokaryote. Yep. So the kingdom prokaryota or Monera or bacteria, whatever you want to call it, are all prokaryotic. Everything else is eukaryotic. That system of classification worked pretty well for a long time. But as we started to develop new technologies, and one of the technologies that was really important was gene sequencing, and particularly genes that encoded ribosomal RNA. And as we started to sequence more and more genes and look at similarities and dissimilarities of gene sequences for different organisms and different kingdoms, we started to see something really interesting. And that was in this particular group right here, these prokaryotes. And they started to find out that there was just an incredible amount of diversity in the basic nature of gene sequences within this kingdom. I mean, all the rest of these gene sequences from all these different kingdoms were very similar with the ribosomal RNAs. And that suggested a fairly close relationship ancestrally. But in this kingdom of prokaryotes, they just found, man, all this diversity of different gene sequences. So a guy by the name of Woese decided that it was time to kind of reevaluate classification systems based upon this new understanding of gene sequencing and what that implied in terms of evolutionary relationship. And he suggested a new system of classification called the three-domain system of classification. And that basically stuck on a higher level of hierarchy above that of kingdom, a more inclusive level of hierarchy. So here's the old Whittaker five kingdom system of classification. So we've got the Monarans, the prokaryotic organisms, and then we've got plants, fungi, animals, and protists. And the idea, at least, is that the ancestral cells first gave rise to these prokaryotic cells, and that eukaryotic life then developed later. Here is the new system of classification, the three-domain system of classification. And here's our ancestral cell line. These were the first cells that evolved. How that happened, we're still not absolutely sure. But very early on, we believe that evolutionarily, these cells started to diverge from one another. And that three kind of basic lines of evolution developed pretty quickly. The three domains are the domain bacteria, the domain archaea, and the domain eukarya. The domain eukarya was then further divided the way the five kingdom system of classification worked into the kingdoms of plant, animal, fungi, and protista. So there's this, in this system of classification, there's this kind of recognition is that there are a whole lot of eukaryotes, but there are a significant number of prokaryotes. Previously, these had been lumped together into one kingdom, Monera, which suggested that they all were, related to one another. The three-domain system of classification system says, these prokaryotes are very fundamentally different from these prokaryotes. Think about the implications of this statement for a second. Let's take a look at a little protozoan like paramecium. And let's take a look at humans, which are in the animal kingdom. According to this system of classification, they are in the same domain. So what that is saying to us is that a paramecium and a human are more evolutionarily related than this type of bacteria is from this bacteria. Because they're different domains. That these are just way fundamentally different than these. We used to lump them together. Than a paramecium is from human. So that shows you kind of the different thinking that this new system of classification brought about. And it was all the result of insights brought about by the application of technology. We're going to spend some time talking about classification. And one of the things that we're going to try to hammer into a little bit is, it is really hard to group organisms, particularly just on the basis of external appearance. And you can come to some really false conclusions about evolutionary relationships just looking at external features. And that's kind of what the five kingdom of classification is. That, here's all these little single-cell prokaryotes. They all pretty much look the same. They must all be related. And they've got to be fundamentally different from paramecium to the cube. When we started analyzing the ribosomal RNA sequences, we started seeing some interesting insights. And evolutionarily, these are much different from one another than anything that is in this particular group. So when we do a brief overview of the classification of microorganisms, we'll do that in a couple of weeks. And it won't seem so brief to you. This is probably one of the more deadly parts of the course, when we try to sort out some of the taxonomy. Keep that in mind, that this new three-domain system of classification has just changed the way we think about the fundamental relationship between different organisms on this planet. That exercise of going through classification systems will introduce you to a very important tool called Berge's Manual. And Berge's Manual is something you will have to become familiar with and facile in the use of, because you're going to be using Berge's Manual in your identification of your microbial unknown throughout the course of the semester. I don't know if you've heard, but in lab we have a semester-long project where in the third week you get this tube of bacteria. They all pretty much look like one another in terms of comparing your tube to somebody else's tube. Might be a little different colors, things like that. But by the end of the semester, you have to identify the genus and the species of your unknown. And everything we do in lab is kind of practice and learning techniques that allow you to do that. It's an exercise that students enjoy. There's a certain amount of, I think, satisfaction of taking this thing you have no idea what it is, and by the end of the semester, hopefully, being able to figure out what it is. So we will be coming back and talking more about three-domain system of classification and the implications of that. Okay, let's go back and dig a little bit more deeply into microorganisms and their relevance and what they do and why they're so important and why I would say life can't exist without microorganisms. And we'll just make a couple of columns, a plus column and a minus column. Plus column will indicate something that microorganisms do that's beneficial. Negative column would indicate something that they do that's harmful. And you've already mentioned one, we can put in the harmful column. And that is disease. Anything else you can think of that microorganisms do that could be put in the harmful column besides making people sick? Yeah. Any of you ever encountered anything that looks like this? Maybe in the back of your refrigerator? The Irish potato famine was a result of this phenomena as well. Decomposition? Yes, honey.
Decomposition, can it be negative or positive?
Good question. Can decomposition be positive? Because, I mean, you can certainly lose a lot of food material, both in the field and then after harvest, once you get things. I don't know how many vegetables we throw out because they get rotten if you don't use them in time. When can decomposition be good?
When you like have a dead animal before?
Yeah, we're going to have a whole list of ecological benefits associated with microbial activities. And one of them is decomposition. Nutrient recycling, is that important in an ecosystem? Absolutely. Without nutrient recycling occurring on a regular basis, life can't exist. One of the things we're going to talk about in a fair amount of detail in regard to nutrient recycling is the nitrogen cycle. And agriculture is absolutely dependent upon the functioning nitrogen cycle. Anybody familiar with the concept of crop rotation? Certain crops remove nitrogen from the soil. Corn does a lot of that. And certain plants add nitrogen to the soil again. What kind of plants add nitrogen to the soil? Legumes, okay, peas, clover, alfalfa, things like that. And why do they do that? Well, if you pull up a legume, you may ever pull up a legume before? What do you see in the roots? They have little nodules. They look like little tumors. And what happens? There are microorganisms living in the roots of those plants. And what the microorganisms do is they convert nitrogen from the gas in the atmosphere, N2, into a usable form of nitrogen, ammonia. Microorganisms have a unique capability of being able to do that. So the plants get the ammonium that they need to make nucleotides and amino acids. The bacteria benefit by this association. What do the bacteria get? They're sitting down there in the root. What does the plant provide for them? Carbohydrate, photosynthesis. Okay. What do you call that relationship with where two organisms benefit one another? It's a type of symbiotic relationship, but there are different types of symbiotic relationships. This one is a you scratch my back, I'll scratch yours, plus plus relationship. That's called a mutualistic relationship. So we'll be talking about this nitrogen cycle and the mutualistic relationship between the legumes and the bacteria. And we will do it in the context of talking about microbial metabolism. How microorganisms produce their energy. And as it turns out, one of the aspects of some microbials' metabolism is its ability to convert N2 gas. N2 gas is very stable, not very reactive, into other compounds like ammonia. That's called nitrogen fixation. So we'll talk about that process. So yeah, this whole nutrient cycling process is an aspect of decomposition, but this is a beneficial one. We've actually taken microorganisms and genetically modified them to become better decomposers. Anybody heard of an example where we've done that? Pardon me? Probiotics gets us into another thing that we'll talk about, Brandon. But new movie coming out. Mark Wahlberg, Horizon. Anybody seen the ads for it? You're all familiar with the Deepwater Horizon oil blowouts in the Gulf of Mexico, BP, all the oil spilled out there. What did they do to try to get rid of some of that oil? They used dispersants, but people argued that just caused it to settle to the bottom. Well, what's a strategy, a technological strategy we use to try to get rid of that oil? We developed strains of genetically engineered microorganisms that could eat the oil-eating bacteria. We have developed genetic strains of bacteria that have been used to clean up nuclear waste spills in places like Hanford, Washington, where there are underground storage tanks filled with nuclear waste. You may know what that process is called, of using genetically engineered microorganisms to try to clean up environmental messes. That's called bioremediation. So that's another beneficial aspect of decomposition. Under this ecological aspect. There's another really, really important one that microorganisms unfairly don't get much credit for. Most of the credit goes to trees. And that is photosynthesis. Photosynthesis is an important ecological process? Absolutely. For a couple of reasons. Obviously, number one, convert light energy into the chemical energy of carbohydrate, which we use by either eating plants or animals that have eaten plants. What's the other important product of photosynthesis besides carbohydrate? Oxygen. No aerobic cellular respiration without oxygen. Most eukaryotic organisms can't survive unless they can respire aerobically. But oxygen has another important function in at least our world today, besides supporting aerobic respiration. And that is in the upper layers of the atmosphere, oxygen's converted into ozone. Why is ozone significant? What does ozone do for us?
As a protection.
Yeah, protects our planet from ultraviolet radiation. Prior to the evolution of photosynthesis, was there oxygen in our atmosphere? Nope. Atmosphere of the planet before the evolution of photosynthesis was a reducing atmosphere filled full of gases like methane, ammonia, but no oxygen. Literally, the 20% oxygen that's in our atmosphere right now accumulated bubble by bubble by bubble as a byproduct of a waste produced during photosynthesis. Where did all life on Earth exist prior to the evolution of photosynthesis? Water. Why? Because water filters out ultraviolet radiation. And ultraviolet radiation is damaging the cells. But as that ozone layer started to form and filter out the UV radiation, what big transition could occur? A transition from aquatic environments to terrestrial environments. Land plants, land animals. It is estimated that 50% of all of the photosynthesis that occurs on this planet is the result of microbial photosynthesis. Now, that includes throwing algae in with microorganisms. So it's not just bacteria. Are there photosynthetic bacteria? Do you know? Yep, certainly are. So another very, very significant ecological function. Well, we'll continue here tomorrow. Okay. We'll finish up this idea of talking a little bit about relevance with some more examples. We'll talk a little bit about where microorganisms occur, and then we will get probably maybe into a little bit at the end of the period talking about some of the history of microbiology. So this is all on that introduction PowerPoint. Okay. So the next slides are pretty much this week on the first PowerPoint.
Yeah.